Hi students, coming to the next class is the structure of the fungi, right? In the last class we discussed about some of the harmful effects and beneficial effects of the fungi. Coming to today's class, we are going to discuss about the structure of the structure of the fungi. See, in the last class we already discussed within the fungi there is a filamentous structures are present, and these filamentous structures certain biochemical reasons not called as filaments, right? These are called as hyphae. These structures are called as hyphae, right? And a group of hyphae are called as mycelium. A group of hyphae are called as mycelium. Mycelium is the major body of the fungi. Mycelium is formed by the group of hyphae. Especially three types of hyphae are present within the fungi, and these three types are combinedly forms the mycelium of the fungi, or main body of the fungi. So within that case, if we discuss about the structure, this is one of the rhizopus. We know about that rhizopus. I thought in the 7th and 8th, 9th, okay, in the earlier classes we already heard about that rhizopus, or otherwise I like to explain in this way. See, for suppose we bought one bread, and if we kept the bread inside the refrigerator, the bread will not be spoiled. Okay, if we kept the bread outside the refrigerator or at the normal room temperature, okay, after 1 week, if we observe the bread structure, there is a black color dotted line structures are there. Upon the white color bread, there is a black color dotted line structures are present. So these dotted line structures are nothing but fungi. The dotted line structures are nothing but the fungi. See, if we take in that black color, okay, pins-like structures or dot-like structures under the glass slide, if we observe under the microscope, especially that structure resembles like this. Exactly that microscopic structure resembles like this. So this is about the exact structure of the rhizopus. So here we are taken rhizopus is the example. Rhizopus belongs to the fungi, and rhizopus is also called pin mold structure. Pin mold structure. This is also called bread mold structure. See, which is grown on bread and which is grown on bread. That's the reason why this is called as bread mold, right? And which is forms the pins-like structures. We know, okay, there is an we are using pins are there, right? This is the pin we are using, right? So here this is the root organ, and this is the and pin and part, and this is the base part. So what I am trying to tell here, this structure, this is the pin structure. So this is the fungi structure. So both these are resemble. Okay, that's the reason why this one is called as pin mold structure. That's the reason why this one is called as pin mold structure. See, mold is nothing but fungi. Here mold is nothing but the fungi. So if we observe the structure of the fungi or the rhizopus, in this case majorly three types of hyphae are present. In the last class also I discussed, right? There is in base part or fingers-like structures are there. Okay, these are the fingers-like structures. See, these are look like roots-like structures. These are look like root-like structures. For suppose one of the bread is there. This is one of the bread slice. This is one of the bread slice. So within the bread slice, some of the structures are penetrated. Some of the fungi structures are penetrated. See, the penetrated structures are nothing but hyphae. Okay, that hyphae is called as rhizoidal hyphae. That hyphae structures are called as rhizoidal hyphae. Remember, rhizoidal hyphae is nothing but within the fungi one type of hyphae which is penetrated into the bread, or which is penetrated into the decaying parts, which is penetrated into the dead organisms, right? So which is penetrated into the parasitic organism, means host cells, right? See, that structures are called as rhizoidal hyphae. So what is the use of rhizoidal hyphae? It's majorly gives the mechanical strength. To that remaining hyphae parts. And remember, whatever that the plants are there, the plants are having the roots. The roots also nothing but the roots penetrating area is called as within the soil. The roots penetrating area is called as rhizopora. So here name also derived from that. Plants having the roots, the spreading area is called as rhizopora, right? Here these also look like roots-like structures, right? And these are involved in the absorption of nutrients, water, mineral, and everything, right? So this part is called as rhizoidal hyphae. And coming to that upper portion, this is the upper portion of the fungi. I already said there is a pins-like structures are present. See, these pins-like structures are nothing but reproductive hyphae. The pins-like structures are nothing but reproductive hyphae. So this is the one of the reproductive hyphae part, and this is one of the rhizoidal part. And these two are connected by the main body hyphae. These two are connected by the main body hyphae. Right? So this is about the types of the hyphae. There is a reproductive hyphae is there, and rhizoidal hyphae is there, and main body hyphae structure is also there. Main body hyphae structure is also there. And these are the three types of hyphae, and these hyphae majorly connected, one on one, finally forms the complete structure of the fungi. So this is one of the rhizoma structure. There is a pin mold, and this one is also called bread mold. Right? So that is about the reproductive hyphae. That is about the hyphae and types of the hyphae. That is about the hyphae and types of the hyphae. That is about complete structure of the rhizomas of the fungi. And coming to the next one. Again, if we discuss about the structure, there is four types are there. There is four types, means here there is a mycelium is there, or this is one of the hyphae structure. Within that hyphae, septas are formed. In certain cases, septas are not formed. So based on that, we are classifying here. Right? So we will discuss about that structure. There is an septated branch, septated branch, and branch, septated branch, and septated branch, and this is septated unbranched. This is septated unbranched. Right? We will discuss now about these four types. So here the first one is that a septated branch. So first of all, I want to know what is septated and what is aseptated. So suppose this is the one of the fungi structure. Right? Here fungi forms the incident species fungi forms the crossroads. So presence of these crossroads. That mycelium is called as septated mycelium. These structures are called as septa. In certain cases, the septas are absent. So this is one of the mycelium. Here septas are absent. So this is aseptated. And this is septated. So based on that, we will discuss about what is septated and what is branched and what is aseptated and what is unbranched. We will discuss now, right? See, the first one is the aseptated branch. So suppose this is one of the mycelium, right? Here septas are absent. Whatever the septas are there, whatever the septors are there, the septors are absent here. So here septors are absent. So that one is acceptated. This is acceptated, right? And branched. Here branches are present. See the difference between unbranched and branched. So this is the mycelium. This is unbranched. This is unbranched. And this is branched. Here branches are present. Here branches are absent. Here branches are absent. And here septors are absent. Here septors are absent. So if the septors are absent here, many number of nucleus are present. Many number of nucleus are present. See, many number of nucleus are present without any septors, right? Without any septors. The condition is called as sinusoidal condition. This is one of the important bit. The condition is called as sinusoidal condition. Say, whatever that mycelium is there, within that mycelium, here septors are absent. This is acceptated, and here branches are present. This one is branched. This one is branched, right? So that is about that one of the acceptated branch, acceptated branch. And remember, within this type, sinusoidal condition is maintained. Sinusoidal condition. Sinusoidal condition is nothing but multi-nucleated condition. Many number of nucleus are present. Many number of nucleus are present. Remember here, cytoplasm is present, cross walls are absent, and many number of nucleus are present. Right? That is about the acceptated and branched. And this is a very simple one. See, within this case, this is the mycelium. This is the mycelium. Here, septors are absent. Here, septors are absent. So, septors are absent, that one is acceptated. Acceptated, right? And next one is branch is also absent. Here, branch also absent. So, that one is called as unbranched. That one is called as unbranched. Acceptated, unbranched. Acceptated, unbranched. And remember here, many number of nucleus are present. Many number of nucleus are present. The condition is called as sinusoidal condition. The condition is called as sinusoidal condition. Remember, within that mycelium there is a septors are absent and branches are absent. So this one is called as acceptated and unbranched. And here sinusoidal condition is maintained. Sinusoidal condition is maintained, right? So that is about the acceptated branch and acceptated unbranched. And coming to the next one is the septated branched. Septated branch means septors are present. So this is one of the this is the septated and branched. So here, this is the mycelium. This is the mycelium structure. Here, branches are present. Branches are present, and cross walls also present. Here, cross walls also present. So, presence of these cross walls. Here, nucleus are separated. Right? So, this is the nucleus. This is the nucleus. This is the nucleus. Right? This is the nucleus. So, this is the sinusoidal condition or not. Okay, first of all, I will explain septated and unbranched. See, here septors are present. This one is the septated, branches are present. So, this one is called as branch. Right? And here septors are present, branches are present. Sinusoidal condition is not present. Sinusoidal condition is not present because here multi-nucleated condition is not maintained. Multi-nucleated condition is not maintained. That is nothing but sinusoidal condition is not maintained. Sinusoidal condition is not maintained. So here sinusoidal condition is not maintained. Septors are present. Branches are present. Branches are present. Then what type of condition is present, sir? You said sinusoidal condition is not present. Then what type of condition this is? See, here one nucleus is separated by the septors. One nucleus is separated by the septors. So the condition is called as monokaryotic condition. Monokaryotic condition. In certain cases, The septa separates a single nucleus. The condition is called as monokaryotic condition. Right? In certain cases, the septa separates the two nucleus. Septa separates the two nucleus. So the condition is called as dikaryotic condition. The condition is called as dikaryotic condition. So remember all of you, presence of septas, either monokaryotic condition, either dikaryotic condition is maintained. Either monokaryotic condition or dikaryotic condition is maintained. Remember, in fungi, presence of septa. In fungi, presence of septa. Mostly dikaryotic condition is maintained. So septated, branched. This is monokaryotic condition. Okay. In certain cases, mostly in fungi, dikaryotic condition is maintained. Right. That is about the septated, branched. And last one, septated, unbranched. See, septated and unbranched is nothing but within the septated and unbranched case. First one is, this is the fungi. Here septas are present. So here septas are present. That one is called as septated. Branches are absent. So this one is unbranched. This is septated. Septas are present, and this is unbranched. In certain cases, only one nucleus is present. In certain cases, one nucleus is present. That one is called as monokaryotic condition. Right? This one is also called, not this one. See, this one, monokaryotic condition. In certain cases, two nuclei are separated. So the condition is called as dikaryotic condition. The condition is called as dikaryotic condition. So simple. Same thing, this one and this one. Here branches are absent. Here branches are present, right? So this is about the fungi and structure. Within the structure, totally 4 types are there, based on the septas and based on the branches. Right? There is a septated branch and a septated unbranched. There is a septated branch and a septated unbranched. See, majority of the cases within the fungi, dikaryotic condition is maintained. Septated and dikaryotic condition is maintained. Right? The next one we will discuss about after that nutrition and after that properties of the fungi. Okay, next one we discuss about the classification of the fungi. That time, okay, sir, in which class or in which type of fungi xenocytic condition is maintained, in which type of fungi dikaryotic condition is maintained. We will discuss, and the examples are also very, very important. Right? Within the fungi, remember the types of the many number of examples are there. Okay, we have to remember all these examples for the examination point of view. Okay, this is one of the little difficult task, but this is not much difficult. Examples are only here difficult. Remaining concept is very, very easy. Okay? That is about that structure and the types of the structures within the